perfect. All right, just let me know when I can start.
Go for it. Everyone, welcome Dr. Lior Goldberg. He's an immunotherapy fellow at City of Hope Comprehensive Cancer Center, pediatric hematology oncology fellow at Children's Hospital LA, and an active Journal Club member. So he's a great guy. Please welcome Dr. Goldberg.
Thank you so much, Rick. Thank you for the kind introduction. Thank you everyone for joining and thank you for having me. You know, I named the talk All Roads Lead to Rome, but it's based on this, you know, work that we had done as a group, and it's actually, you know, I find this work really cool. And Eric and me were kind of like the leading investigators on this project. So I'm going to thank Eric at the end, but once again, thank you, Eric, for giving me the opportunity to collaborate together. No disclosures. All right, so I think, you know, our story starts with T-cell trafficking, right? So we all understand that adaptive immune response requires some cellular plasticity, and the plasticity is very wide. It has to do with the fact that, you know, for example, T cells have different subsets, you know, whether you're a naive T cell or an effector T cell, you have plasticity that is required to traffic, to leave the bloodstream and transmigrate into different tissues. And you have, for example, plasticity that is related to metabolism, right? Because you have to adapt to a microenvironment, right? But the whole process is really interesting because, you know, we go into the details in the next slide. There is a very interesting interaction between the T cells and the vasculature in order to promote trafficking and transmigration. Okay. The other thing, in the context of CAR T cells, we know that CAR T cells perform their anti-tumor activity both in medullary and extramedullary sites. And this is kind of like their biggest advantage, right? They control systemic disease as well as medullary disease. When you look at the process, so here's the leukocyte, right? The T cell or the B cell, in our case, the T cell, there is a multi-step cascade that leads to this transmigration, because T cells, you know, it depends on the kind of T cell, but they flow in the blood or in the lymph node. They have the regions of interest, but then they get a cue and they start this multi-step cascade that leads to, at the end, to a transmigration. And that was our starting point. We looked at the literature and we were very intrigued by the fact that we all know that CAR T cells can, you know, perform anti-tumor activity both in medullary and extramedullary sites, but what is the biology behind it, right? So based on this, you know, trafficking molecules, we designed a very cool panel. So everything that is on the that is marked on the blue color is related to T cell lineages, and we have very nice thing in our cars. They all express a truncated EGFR. So if we gate on the EGFR, and I'll show it later, we can identify T cells. Okay. So the blue is the cell lineages. The yellow is related to activation and maturation. Everything in the gray color is related to trafficking. And then we have the immune checkpoints. Now, one thing to keep in mind is that CARs and, you know, CARs and regular T cells, or CAR T cells and regular T cells are very different species. Okay, because first, you know, when we take a patient and we want to give them CAR T cells, we have to leukapheresis the T cells and then expand them. And in order to expand them, we ex vivo activate, we stimulate them, we transduce them. And that is a very, very robust process. Okay, just think about it. You know, these patients that get these CAR T cells are you know, heavily treated with chemotherapy. These T cells, you know, are heavily treated, have been exposed to multi-agents, and we usually, you know, these are relapse patients. we give them bridging chemotherapy and try to pick the right moment to leukapheresis them and collect the T cells. So we start with a very low number of T cells, but also their quality, given the fact that they were exposed to so many chemotherapy lines of treatment, is, it is questionable, right? And then patients are preconditioned with lymphodepletion in order to encourage the CAR T cells expansion. And the most important part is that CAR T cells are independent of the MHC, right? So regular T cells are, in order to be activated, they need an MHC signal, and CAR T cells are independent of the MHC. Now, when you look at the challenges to enhance CAR T cell efficacy, we have many challenges, right? One is of course, the impaired T cell trafficking, right? Either going to the brain or into a solid tumor or a hostile microenvironment. The impaired T cell function, T cells that become exhausted, poor T cell expression and persistence, and of course the tumor heterogeneity, right? So most of this talk, we're going to talk about trafficking and function. We won't be talking much about persistence, or we won't be discussing the tumor sites though. When we want to investigate CAR T cells, we have quite a few dilemmas, right? So, first we need to decide what should we do? Should we use the clinical samples or should we use a preclinical model? The other one, how do we detect CAR T cells? The biggest challenge, and I think this is really the biggest challenge for the field, is the low number of CAR T cells, right? So, if you take a blood sample on day 7, which is the peak of expansion, you have, roughly a few thousands of, CAR T cells on a, 1 to 2 ml of blood. But if you go to day 28, when the T cells contracted, there are very few T cells in the bone marrow and blood in the CSF. So the low number of CAR T cells is, I think, probably the biggest challenge. Then you have to ask yourself, what do you want to investigate, DNA versus RNA versus protein? And of course, we are, interested in cytoph, so we decided with proteins. And then we all know that flow is limited by the number of proteins, and not preaching to the choir, but the answer comes from cytoph. So how to detect the CAR T cells? As I mentioned, we have egfr on our CAR T cells. So whatever is CD3 egfr positive would be CAR T cells, and you can see it's very nicely gated and then separated to CD4 and CD8 and on and on. Now the other thing is, of course, what we need to take into consideration is to have how to design the panel, and there's always the issues of conjugation, spillover, the sensitivity of the different metals based on their mass. And of course, like any other good experiment, you need to do calibration and titration. And the question that we started with was, can we detect CAR T cells with very low abundance? Can we detect 100 CAR T cells, 500 CAR T cells, and so on? And we managed to do so. So the first thing we wanted to do is to better understand our model. And what we started with is we took healthy donors T cells. So, and we insisted doing that on a ultra fresh sample. So it's healthy donors, they come to the blood bank to donate blood, and then we get the leftovers, and we cycle the leftovers, and we ran cytometry the same day. And the reason why we insisted doing ultra fresh blood is because cycles of freezing and thawing cells can affect some of the markers. For example, we know that CD62L is being, and CCR7 are both being affected by freezing and thawing. So you can see here how nice we gated on the 6 major subsets. So T-naive, stem cell memory, central memory, effector memory, and TEMRA on both CD8 and CD4 T cells. And you can see how powerful this single cell analysis, because, we found all this like bimodal distribution. Usually, if you do a Western blot or, even traditional flow, people, would show the median expression, right? Either through a heat map or whatever, right? But, this is beautiful because it just tells you that it's actually a spectrum of cells in a group or in a marker, right? So, just to kind of like illustrate, if you look at granzyme B, as the T cells mature, these are healthy donors T cells, you can see as they become more cytotoxic as they mature from the naive state to the more effector state, the granzyme B goes up, right? Which makes sense. Intrigued by our findings, we asked ourselves, okay, so, we started by gating on, the major subsets, but can we identify, cells that were undefined, right? So here you can see the different subsets and the black islands. This is supervised clustering. Those were the ungated cells. And then we did unsupervised clustering, which kind of like revealed what are these ungated cells are. So you can see different 6 clusters, right? And when you look at their lineages and their phenotypic markers, you can see that 4 out of them are what's called double negative T cells. We all have double negative T cells, up to 2.5% in our body. If you have more than 2.5%, depending on the clinical context, you have something called ALPS, autoimmune lymphoproliferative syndrome. And 1 cluster was a double positive, and we all have double positive cells in our body. But we found a new population, which had a memory marker, memory markers, not just memory marker, which just shows how powerful, Cyto and this data is. And then we wanted to investigate the longitudinal plasticity, right? So just temporal changes. So we took 3 healthy donors, we took their PMBCs, and we just produced CARs as we do for patients, right? So you stimulate them with beads, you transduce them with a lentivirus, the CAR, and then you remove on the beads, and on day 14 you have a CAR. But in order to mimic CD19 engagement, we gave them artificial presenting cells with CD19 beads, and we looked 1 day post-stimulation and then 1 week post-stimulation, okay? To get a better understanding about the temporal changes. So you can see, for example, when you look at the granzyme B, this so the first violin plot is PMBCs, and then it's the product, day 1 post-stimulation, day 7 post-stimulation. So for example, when you look at granzyme B, the leukophorese T cells are resting T cells, product is highly active, but then 1 day post-stimulation, even higher cytotoxic activity, and then day 7, it starts to decline, or it is declining. We didn't follow more than day 7, but at some point it returns to baseline, right? So it just gives you once again a view regarding the temporal changes. But what was really intriguing is when you look at the trafficking molecules. So for example, look at PSGL. So these are the PMBCs. These are the products, right? So you can see that many of the trafficking molecules in the products are highly expressed, which raises a question: are we, by just producing CAR T cells, priming the T cells to migrate by overexpressing all these trafficking molecules, right? Kind of like priming them to migrate and fight. Once again, you can see, for example, CD69, which is a marker of activation and tissue residence, has a bimodal distribution by day 7. The next thing we did is we used t-SNP plots, and we did unsupervised clustering, and look at the plasticity. These are the PMBCs. Look at the product, how different it is from the PMBCs. Look at day 1, day 7, a new cluster, right? So it just shows a very important concept regarding CAR T cells, that they are not locked. They have their own temporal plasticity in this case, right? And you can see how the clusters change over time and the abundance, and there's a heat map of the different clusters. And I won't go into the details, just to show kind of the concept, right? So this gives us the proof that there is temporal plasticity. But then we wanted to look at spatiotemporal plasticity, right? So what we did is we look at the patient samples. So we took the leukophorese T cells from the patients. It's all the same. All samples were for the same patient, right? So we took the leukophorese T cells, the product, the peripheral blood on day 7, day 28, the CSF on day 7, 28, and the bone marrow on day 28. So it gives you a wider scope, spatiotemporal scope, right? And here, once again, these are the leukapheresis T cells, CAR product, peripheral blood day 7, peripheral blood day 28, bone marrow day 28, CSF. Once again, look at the trafficking molecules. Look at the products. Just by producing products for patients, we upregulate these trafficking molecules, possibly priming the CAR T cells to migrate. Now, what's really nice is, for example, when you look at CXCR4, which is a homing molecule for hematopoietic stem cells, to the bone marrow, you can see a bimodal distribution. So would that suggest that there is a subset of CAR T cells? They have high CXCR4 and they have a tropism to the bone marrow, right? And there's a lot of data here. I don't want to go to the nitty-gritty details because it's a plethora of information. Now, when you look at the plasticity of the CAR T cells, once again, look at the leukapheresis T cells, the product. Look at day 7, day 28, blood, bone marrow, and CSF. And you can see how the plasticity changes over time and space, which is amazing. These are all patient samples, right? Not mice, not in vitro, real life experience. Now, when you look at the different clusters, you can see that some clusters are enriched in, some of the locations. For example, when you look at cluster 3 and 4, they are highly enriched in the CSF, right? When you look at cluster 1 and 2, they are highly enriched in the leukapheresis T cells. Cluster 9 is highly enriched in the product, right? But how can you learn more about these clusters? So what we did is we used something called Shannon Diversity Index and Simpson Diversity Index. So Shannon gives you the repertoire, how diverse the T cells are, right? So when you take leukapheresis T cells, they are very diverse, right? When you produce a product, you enrich for a certain population. So the diversity decreases. But I think this is just beautiful. Look what happens over time. The diversity is being regained almost to the level of the leukapheresis T cells, right? And the Simpson gives you a measurement of how even the distribution in a group. So the leukapheresis T cells are evenly distributed. There are many groups inside the leukapheresis T cells, but the product is dominated. It's uneven, and once again, as time goes by, this unevenness changes to a more even distribution. And I think this is beautiful. The next thing we ask ourselves is about the CSF, right? Because CSF has a critical role for leukemic and lymphoma patients. It's what we call a hiding site. We have a lot of relapses related to the CNS, and we ask ourselves, what these features of the CAR T cells in the CSF, are they impacted by the underlying diagnosis, by the CNS status, or by the time? And not. These CAR T cells had a very unique features that were regardless of the patient's disease, CNS status, or sampling time. And you can see that they're almost homogeneous. At the end, what we did was kind of like circle back to our initial observation, and we looked at the healthy donor subsets, and we asked ourselves, can we correlate between the different CAR T cells and the subsets, the healthy donor subsets? So you can see, for example, that the leukapheresis T cells are kind of like evenly distributed. The product may be a little bit more effector shift. Peripheral blood day 7, more effector. Peripheral blood day 28, by far more effector. But you can see, for example, in the CSF, there is enrichment of stem cell memory cells, which might suggest that this is a memory niche for the CAR T cells, residence niche. So to summarize our findings, we all know that adaptive T cells immune response requires cellular plasticity, and the studies of CAR T cells have primarily focused on very limited number of phenotypic markers in the blood. And that, of course, represents an incomplete view of the CAR T cells complexity. And this is the first paper showing data on bone marrow and CNS. And we used Cytof to simultaneously analyze trafficking and functional proteins in patients across the time and space. We revealed this spatiotemporal plasticity, and we showed that patients' CAR T cell product showed increased expression of trafficking and activation molecules. We saw these spatiotemporal alterations and very distinct signature in the CSF. And these data provide potential framework to remodel CAR T cells and enhance immunotherapy efficacy, right? Because now that you understand how they traffic, you can enhance their trafficking molecules in order to get better tropism. That's what we are doing right now. We have several projects based on this paper to enhance tropism. So I want to thank my my PIs and my lab members, my colleagues, Dr. Foreman, Dr. Wang, Dr. Ryan Urek, and Vibhuti Vyas, and of course the one and only Eric. I think this work, is a true scientific collaboration between Eric and us here at City of Hope, and all of this wonderful data is a true testimony of Eric's research. The guy is a genius. And I want to thank Connie Inlay from Fluidim that helped us to start this project. Once again, Connie was so helpful and supported us throughout this project. And I think the number of times that I texted Connie at 10 o'clock in the evening about stuff, well, I won't disclose, but many times. So thank you, everyone. Thank you, Connie. Thank you, Eric, to our City of Hope researchers. And I'll be more than happy to take questions.
Thank you, Lior. All right, everybody, feel free to ask questions.
I have a question. Dr. Goldberg, great work. My name is JC Villas-Boas. I'm a hematologist here at Mayo Clinic. One is a more technical question regarding panel design for your experiment. Because you're evaluating all markers in the same population, you probably don't have the ability to distribute mutually exclusive markers in other populations, which is typically done with Cytof panel design. So my question: did you feel the need to use any sort of normalization, compensation, I should say, for this panel, or was it not necessary?
It wasn't necessary. The only thing I have to say is that, when we calibrated our system, we calibrated it to 1 million cells. So for example, if you have a CSF sample with 2,000 CAR T cells, right? And you have kind of calibrated your system for 1 million cells, what we did to keep this persistent 1 million cells per sample starting point, we spiked in regular T cells that do not express EGFR. And that was, that was brilliant by the sense that we didn't see any batch effect. So that was, that was the kind of like the only obstacle regarding that point. The other things, when we calibrated the system initially, so of course we looked at spillover. We used the panel designer, and then we ran a few samples of LT donors, and we looked at the spillover with a specific pattern that we all know. This stretched line that goes from the left lower quadrant to the right upper quadrant, that is very suggestive of spillover. But I think at the end, looking back, and thanks to the help of Connie and Eric when we designed the panel up front, I think for this panel specifically, there was almost no spillover at all. It was like really nothing. We have some other projects, and I'll be more than happy to share in in the near future even more complex panels. And in these panels, we had to deal a little bit with spillover, but once again, not something too too significant. I think this is this is one of the biggest advantages of Cytof versus flow, right? Flow, you're so limited with the number of proteins. You have to calibrate, compensate, still spill over. There is, it's as time goes by, when I kind of like, and some of these samples, interestingly, some of these samples, we had flow data because we do our QC. And I looked at the flow samples and I looked at our Cytof data. It's uncomparable. The flow is like, really, it's low grade when you compare it to the Cytof.
Got it. Perhaps I could ask a follow-up question regarding exactly that, the fact that, the number of cells in your samples are low to begin with. You at day 7 at a maximum peak expansion, you may have a few thousand T cells. And you put that through a Cytof, you're probably going to have in a good day 500,000, half of that back in your data side, right? So when you're doing your clustering, did that influence the choice of clustering algorithm to be able to detect rare events versus not?
So we were, and this is a great question. I think this is the most fundamental question, that needs to be asked when we deal with CAR T cells. Once again, the number of CAR T cells is very low, especially, post day 7, day 28, and so on, right? So I'll answer this question in 2 parts. The first part is this panel specifically, which looked at very few intracellular markers, was granzyme B and interferon gamma, went through mild permeabilization, okay? And we lost less cells because, once you go into panels that deal with transcription factors or proteins that are in the nucleus, you have to do a very strong permeabilization and fixation, and you lose so many cells. So one of the things to consider is how many cells you're going to lose through the process. We got to a point that we roughly lose around 20% of cells from the starting number. And for all of our samples, we actually had to down sample, and we analyzed 5,000 cells per sample, 5,000 pure CAR T cells. We had a very few samples that we went down to 3,700. But once again, I think anything above 500, you are golden. Like, it's single cell proteomics, it's very robust. Anything about 500, you're golden. We have another project that we have, unfortunately, as part of that project, we have so many proteins that are intracellular, but deep in the nucleus, right? And we have to do such a harsh permeabilization and fixation, and there we lose 50% of the cells. So if you have a clinical sample that you start with 5,000 CAR T cells, at the end when you acquire the samples in the core or Cytof, you start with 2,500 cells, and then when you start to shoot the cells to the nebulizer, you end up with 500 cells. But I think as long as you're above 500 cells, you should be golden.
Thank you.
I'll jump in with some big picture questions. So I'm from Fluidim, so I'm already sold on the technology, obviously. But if you could just sum up 2 things. So what was your biggest challenge with this project? And then you've already kind of mentioned it, but why did you choose Cytof for it?
Right. So I think the biggest challenge was, the processing, making sure that we don't lose too many cells. Right. So, there is a learning curve, right? Like anything in life, right? So there's a learning curve. And I think, for this panel specifically, after, 2-3 times when we kind of like dealt with regular T cells, we all felt comfortable, moving forward to the clinical samples that are so precious. So I think, before moving forward to dealing with precious samples, I suggest everyone to take the time, run it a few times on just, regular T cells or whatever, you're like, control samples that are not precious, and optimize the system. So you feel comfortable, running your clinical samples because they are so precious, right? So I think the biggest concern was like, can we detect the CAR T cells, giving their low number, and would the egfr work? But the egfr, we took a few products from healthy donors and we saw, instantly that the detection is good. And we can detect the CAR T cells. And I think the biggest advantage of doing the Cytof is it truly gives an in-depth understanding, or it gives you a tool to answer a biological question. We had a very specific biological question. How do CAR T cells traffic to all these locations? And, if you think about flow, even single cell RNA, right? So, we all know that proteins are not equivalent to RNA, right? So even if you do a single cell RNA, it doesn't answer the question at the protein level, right? Might give you more data, but at the end, you would still need to add the second layer of the protein on top of the RNA if you choose, RNA as screening or whatever. So I think, if you want to answer a very specific biological question that you can define, I think Cytov is a great tool. I wouldn't use Cytov for massive screening because you're still limited by the number of proteins, right? And, sometimes in biology, pathways can be made up of like 100 proteins or 200 proteins, right? Or they interfere with, many partners. But if you can define your biological question and you can limit your search to the degree of what Cytov offers, you're golden.
Thanks.
One more question, Dr. Goldberg. Does your EGFR antibody work in formalin-fixed paraffin-embedded tissue? Can you see the T cells in the tissue itself?
I haven't tried it with the, with the Cytov for biopsies with the Hyperion system, but based on our experience with immunohistochemistry, it works. It works very well. Yeah, we can use the EGFR and it works very well. Yeah, and I assume it should work the same with Cytov. We have a project that we are studying now, looking at bone marrow samples, kind of like looking at the microenvironment of CAR T cells, and we had a kind of like a pilot, and the EGFR is being expressed, and you can detect the CAR T cells with the EGFR. Now, one thing I do want to mention is that we are very privileged because of all of our CARs for B-cell malignancies have EGFR, but some of the other CARs for other diseases do not have EGFR, right? Even if you look at Kymriah for Novartis, they don't have they don't have a protein that allows you to track CAR T cells, and there have been antibodies for the CAR. I can tell from my experience that currently they are not as powerful as the EGFR. So the detection level is by far lower than the EGFR. It has to do with the CAR that changes its expression over time. Okay, so once again, if you wanna start using clinical samples, precious samples, and you don't have the privilege of having EGFR, and you want to get on the CAR itself, what I would suggest to do is to take a product and start to stimulate it and give it a month in vitro, and then 1 month later to look at the CAR excretion. Okay, because the product, they get the beads, they get the cytokines, everything is so heavily upregulated, so you get a very good detection of the CAR. But as time goes by and the lymphodepletion effect decreases, vanishes, right? Then you don't have all the stimulation to the CAR, and the CAR can have very low expression. So before moving forward with clinical samples, if you do want to gate on the CAR specifically, do a prolonged in vitro assay and make sure that you can detect the CAR T cells, day 30, day 60, because clinical samples, it's I don't need to say, but these patients went through a bone marrow biopsy, they went through a lumbar puncture. These are invasive procedures, and we were very privileged to have these samples. So we should respect that.
Absolutely. The Acro Biosciences has a good anti-angiotype anti-CAR antibody that I've tested. It's very expensive, but it's very good too.
Okay. And is it conjugated with a fluorophore for a flow, or is it like just a purified antibody you can conjugate to heavy metal?
They do sell it conjugated to PE if you want. I bought the primary and conjugated for Cytov. Okay,
yeah, perfect. And it works? Like, have you seen like a good detection, 30 days, 60 days post-infusion?
I only did it in vitro using a spike-in CAR on a peripheral blood sample, and the signal is very strong though.
Yeah, Yeah. So the only that sounds promising. The only thing I would just make sure that like it persists. Day 30. I can share with you guys. We looked in our flow on our clinical samples, and we can see detection. We can detect the CAR, but as time goes by, the signal, the protein decreases, and you get less detection of the CAR, and then it's kind of like a gray zone. What do you want to get in? What do you want to get out? Right? You don't want to get cells that are not truly CAR. And also, what does it mean, right? Let's say you have a T cell, the CAR T cell, that has a very, very weak expression of the CAR. What does it say about the CAR? Maybe it's a different population, maybe it's a dying population, or whatever, right?
Yeah, hi, this is John. I'm actually from Slingshot Bio. I'm actually crushing this journal club, but very compelling, good talk. I just want to quickly go back with the uptick in trafficking, how it relates to cell exhaustion. I don't know if that was covered or if you have gone over it, but again, if you made just general correlations, my thought is that if there is immediate, increased activity in trafficking, does it mean that these cells are actually doing their job, and ultimately, like, how does that correlate with exhaustion? So maybe any insight there would be helpful.
So for all of these patients that we used, they all achieved complete remission by day 28 disease evaluation for the bone marrow. So the bone marrow cells were not exhausted, and we didn't see significant, expression of PD-1 or TIM-1, which correlated or did not correlate, inversely correlated to the complete remission. But one thing that we need to take into account when we talk about CAR T cells exhaustion is that all these markers, whatever you choose, PD-1, TIM-3, LEG, right? It depends on the context, right? Because there are also activation markers. So you can see, T cells following activation increase in the expression of some of these markers. So I think it's an excellent question. I think we need to better understand the context, mainly the clinical context. So having a huge cohort which haven't achieved remission, and look at their CAR T cells. For this study, it wasn't our scientific question. In all of our patients achieved complete remission, and we didn't see significant upregulation of the exhaustion markers. But in a large cohort, dissecting responders to non-responders, it would be an interesting question. But also giving the caveat that some of these immune checkpoint proteins can be upregulated just by activation.
Well, thank you.
Hi, Lior. Great presentation. I had a question on in your trafficking screening. Did you look at? I know you looked at beta 7, and I believe the CD49 ALK4 was looked at. Did you look at CCR9 expression? I
didn't look at CCR9. So it's interesting. It's a good question because we kind of like we had a biological question. We looked at the literature. There was no literature on CAR T cells trafficking, so we kind of like focus on the major mediators, right? Major proteins. But there's a whole group of other proteins that needs to be added now, like you're suggesting. So I think now that we have proved the concept, I think you're right. The next step would be to, expand this to more markers, right?
Yeah, and that was kind of the other question I had in my mind. I don't think you have an answer right now. Is if there's any preferential trafficking to different tissue sites, or is it just kind of a general dissemination systemically?
No, we saw a different signature, especially in the CNS. Yeah, the CNS had a very unique features. And once again, when you look at CXCR4 in the bone marrow, for example, you can see this bimodal distribution, which tells you, okay, there are CAR T cells that highly express CXCR4. Maybe they are the one that would persist, right, in the bone marrow, because we all know, right, we give mosibid in order to mobilize hematopoietic stem cells when we collect hematopoietic stem cells, right, which is CXCR4 antagonist. So I think when you look at the small details, you can see clues, and differences between the different locations.
Thank you.
Eric, would you like to add anything? I'm sorry, I like you know, once again, as I mentioned, you know, this project was done by Eric and me, and as I shared with you guys, Eric is a genius. But you know, all of the analysis that we shared with you guys today was done by Eric and me, and you know, we are extremely thankful. And this is really the first and only study that looked into this, you know, biological question.
No, I think you summarized everything well, Lior. Thank you very much. Like you said, it's a privilege to work with these samples. I thought that was, for me personally, working on this project, one of the really cool aspects was that all these patient samples had, these patients where these samples are from rather, had achieved complete remission in the time that we're looking at. So it was really a privilege to be able to analyze them. But yeah, I'm interested in looking too at some of the responder versus non-responder type changes and see what we may be able to see there. And potentially there's some of these markers we found differentially expressed here are kind of missing or lacking from some non-responders. So it'd be interesting.
Right, yeah, that's our next step. Pending, cooking. I hope, you know, I hope that in the near future, Eric and me are doing some more projects. We are looking at, you know, CAR T-cell metabolism, and we're looking at other aspects of CAR T-cells, and I hope that soon we can share with the group some more cool stuff that we are doing. Really, you know, I think this project was kind of like the starting of a longer journey.
Absolutely. Well, if there aren't any more questions, I think we can wrap it up. That was great, Lior.
Thank you.
Great presentation. Like I said, looking forward to that journey and helping discover new pathways and mechanisms of CARs.
Right. Thank you, everyone. Have a good day.
Thank you. Thanks for the invite, too.
Thanks, Lior. Great job. Thanks, Eric.
Thank you.
You all.
Thanks, Lior.
Hey, Connie.